Welcome to Ask and Learn.
Hello students, in this question we have been asked various questions. The first is about the structure of lipid in egg yolk. So egg yolk lipids are primarily consisting of triglycerides, phospholipids and cholesterol. Triglycerides consist of glycerol backbone with 3 fatty acid chains attached, while phospholipids have a glycerol backbone with 2 fatty acid chain and phosphate group. Cholesterol is a sterol with 4 ring structure and a hydroxyl group. Types of lipid present in the yolk based on polarity. The two main types based on polarity is polar lipid and non-polar lipid. Polar lipids include phospholipids, phosphatidylcholine and phosphatidyl ethanolamine, and glycolipids, while non-polar include triglycerides and cholesterol. Here we have phospholipids. Next, we are talking about Lieberman-Burchard test principle. The test is a color reaction test that is used to detect the presence of cholesterol and other sterols. The test involves treating the lipid sample with acetic anhydride and concentrated sulfuric acid. The reaction produces a color change, typically from blue to green, indicating the presence of sterol. The importance of each reagent is as follows. Acetic anhydride acts as a dehydrating agent. Dehydrating agent promotes the formation of colored complex. Concentrated sulfuric acid acts as a catalyst and provides the necessary acidic environment for the reaction. Differentiate between thin layer chromatography and column chromatography. Separation of components in TLC. The separation occurs on a thin layer of stationary phase coated on a glass or a plastic plate, while in column chromatography the separation occurs in a column packed with stationary phase. Solvent migration in TLC, the solvent migrates vertically up the plate due to capillary reaction, while the column chromatography has the mobile phase, the solvent moving down according to the gravity and external pressure. The type of support used, TLC uses a solid support like silica gel or alumina coated on a glass, while column uses a packed column with solid support like silica gel or alumina. Purpose of using ninhydrin and iodine in TLC. Ninhydrin is used to detect the presence of amino acid and amines on the plate. When reacted, these compounds produce a purple color. Iodine is used to stain agents to visualize non-polar lipids on the TLC. It forms brown spots that can be observed. Comparing the results for TLC and qualitative tests, the results obtained from TLC and qualitative tests should agree with one another, as both methods are used to identify and separate lipid components. TLC is a visual representation of the separated lipids, while qualitative tests such as the Lieberman-Burchard test confirms the presence of specific lipid types. If the results do not agree, it may indicate an error in the experimental procedure or maybe interpretation of the results. So these are your answers. I hope you found this video useful. Thank you.